The primary goal of cell membrane is effectively to create a molecular barrier so that things don't randomly and spontaneously diffuse out or diffuse inside the cell. And that's what keeps a cell healthy. In the cellular membranes, multiple things regulates their function. So if a purpose of something to make sure a neurotransmitter comes in within the neuron, there has to be other set of proteins that regulates when they should come in or when they should not come in. And my lab's overarching goal is to kind of develop quantitative technologies where we can actually understand how these things come together in the cellular membrane at the right time, at the right point, at the right place, and then how do they go away? My lab's research is to really understand membrane proteins. To understand that, you need a variety of techniques and tools in a multidisciplinary approach. So for example, if you want to understand a protein X and where they meet their molecular friends and neighbors, you need to have a nanoscopic view where you can home into that region of a membrane where those interactions are taking place. So that's where some of our synthetic chemistry tools and chemical biology tools come in, where we develop approaches that allows us to effectively cookie cut the membrane into small segments where we can really target into a particular region of the membrane where a target protein, where a specific interactions are taking place. Now, when you have done that, then you need molecular resolution to understand what are the molecules we have here. So that's where we use a lot of mass spectrometry. And all of that effectively gives you almost a pixel by pixel information about what's the design principle of the cellular membranes that regulates its function and its ability to communicate with surroundings. Our hope is that this allows us to basically develop a nanoscopic view of effectively how the cellular membranes are designed so that they can respond to specific stimuli and what happens in healthy state, but also what happens when they're not healthy. When you're solving something that doesn't just solve one question or one protein or one pathway from neuronal signaling to insulin secretion, but looks at something that is an overarching theme in both human health and disease, then that gives you that added motivation that it's not just going to illuminate the questions that we are interested in solving within the lab, but it can be spread across the aisle, across my colleagues, and even people we don't know that can be adopted to solve questions that I don't even know exist.